Hey guys, Mr. Arnold again. We're going to talk in this video about DNA replication. We discussed in class sort of a little bit about the structure of DNA, and you're working on your DNA projects to show that structure. So remember that what the structure is that we've got a sugar phosphate backbone, okay, and so that's this area here. Okay, comprised of sugars and phosphate. That's why we call it a sugar phosphate backbone. Okay, so we've got phosphates here, and then we've got these little pentagons right here. These are the sugars. In the case of DNA, remember that's deoxyribose. And then in the middle, what you have are the base pairs linked up. And if you remember correctly, our base pairs always have to pair up in the same way. So you've got C always has to pair with G, and T always has to pair with A. Okay, and so you've got adenine and thymine, okay, or thymine and adenine, and then you've got guanine and cytosine, or cytosine and guanine. It doesn't, they can be on either side, but they always have to pair up with one another. Okay, so what is replication? So replication actually is the DNA getting copied. Now, if you remember back to our processes of mitosis, that this is one of the things that happens, that we have to have replication occurring. The DNA has to copy itself so that when we split the cells up, that we've got two cells that have identical DNA. What we've discovered over the course of time, as far as the method for this, is that DNA undergoes what we call semi-conservative replication. Now, what does that mean? What does semi-conservative mean? Semi-conservative means that you have the original strand of DNA here. Okay, so this is the original strand, and then it gets broken up, or it gets unwound basically, and we're going to talk about that. And then what happens is that it gets unwound and then new base pairs get attached. And so we call it semi-conservative because you still have one of the original strands in each of the new strands. If you look down here at the bottom, I've essentially got two new strands of DNA, and that original blue strand there's half of it basically in each of the new strands. So it's semi-conservative in that half of the original gets conserved and then half is made new each time we replicate it, okay? Rather than making a whole new strand of DNA, each new strand has half of the original strand, and that's the semi-conservative part. So this is a good little diagram for how this occurs. Now you may be looking at this going, oh my gosh, there's a whole lot of things here. Don't panic. We're going to walk through very carefully each one of these things to see what's going on. But I'm going to give you a quick overview, and then we're going to talk about each specific thing. So this is the way that the DNA originally is. It's curled up in its double helix. It gets untwisted, basically. It gets what we call unzipped. Okay, so these are the base pairs here in the middle. Those base pairs get disconnected from each other, and now you have two strands, one on the bottom here, one on the top. And then each of those two strands is going to have the base pairs rematched on it. Okay, so these are new base pairs being rematched. These are new base pairs here being rematched. And so then at the end, we're going to have two new strands of DNA. Now, also remember that we had a 3' and a 5' end of the DNA. That's going to come into play a little bit. And if you don't remember what all that is, jump back to that DNA video that we watched before this, and that will help you out a little bit. So let's talk about the actual steps in this replication process. So the first thing that happens is that the DNA gets uncoiled. So right here in its original form, it's in its double helix. And so to be able to do the copying, we have to basically uncurl it. And to do that, there's an enzyme called isomerase or topoisomerase that basically is used to relax the DNA to uncoil it. So it's basically just in its ladder form now. Okay? So that's the first part. The second part, and this is the part where a lot of people consider to be where it really gets down to the replication part, is that you've got helicase is the second enzyme here. And this helicase, that's this blue triangle here. Okay, that's the helicase. And what it is going to do is it's going to break apart the base pairs. Remember that the base pairs are bonded together in the middle. So you got cytosine and guanine, you've got adenine and thymine. And what helicase does is it basically comes in between them and breaks apart the bond between each base pair. And so what you end up with then is one base pair, okay, on each side. So you've got two of the old strands of DNA, two of the different sides. And those two strands each have one of the base pairs, one of the nitrogenous bases. The other pair is on the other half. So what was paired and bonded together is now split apart by the helicase. Now, we're going to talk first about what happens on this end because it's a little bit more complicated up here, and we'll talk about that in a second. So what then happens is that an enzyme, and this is probably the most important enzyme, helicase, topoisomerase, those are important, but helicase and DNA polymerase are the really important ones. The DNA polymerase, that's this sort of rectangular box here, and of course these are just representations. They're not actually triangular or rectangular or anything like that. Way more complicated than that. But the DNA polymerase is going to come through, and it's basically going to take new base pairs and match them up with the ones that are left. So this is the original link here. Okay, this is the original strand of DNA. And then new base pairs are going to get formed with a new phosphate sugar backbone on the other side, so that now over here we've got two strands again. Okay, and then of course that will get rewound back up to form the actual DNA that we're sort of used to. Okay, so how does that actually work? Well, if you look at this diagram, this is just sort of, we're zooming into that area with the DNA polymerase. We're going to say that DNA polymerase is this box here. And so this is our original strand of DNA over here. Okay, remember it got broken too, so this is one of the strands, and this is its code. Okay, so adenine, adenine, guanine, thymine, cytosine, and so all of those, right? Okay. And so what the DNA polymerase is going to do is it's going to grab another base pair, okay, the matching base pair, and it's going to take that base pair and it's going to line it up with the correct part of the code, okay? So it reads, this is a cytosine, the only thing that can bond with the cytosine is a guanine, and so I'm going to match that up, and this sort of all rolls through here, okay? And the whole thing, if we grab the whole thing and just slide it through, it's going to slide through almost like a ribbon, through the DNA polymerase, and at each point the DNA polymerase is going to match up the appropriate base pair to it. And it's easy to tell, and this works beautifully because there's only one possibility. If this is a C, then what matches has to be a G. This is a T, so then what's going to have to match here? Exactly, that's going to have to be an A, and that's going to have to be a C, and that's going to have to be a T, and a T, and so it is going to be able to tell because nothing else will line up there, right? Nothing else is going to match in there. So DNA polymerase is extremely accurate. It actually, there are errors, and we'll talk about the mutation part a little bit later, but it doesn't happen very much. This is an exceedingly accurate enzyme, and it's because of the system. It always has to be C and G. It always has to be A and T, so the matching is relatively easy. What I didn't mention before is that you may have noticed these little purple nodes here. Once the DNA unwinds, you have to hold them unwound because the tendency would be for them to bond back together and then to rewind again. But so you've got to have something that sort of holds them apart, and that's what these, what we call SSB proteins, or single strand binding proteins. And you're like, that sounds really complicated. Single strand, okay, it's one strand now, right? And it's binding them. It's just holding them so that they hold apart so that they can then run through the DNA polymerase. Now, you may have noticed that all we talked about here was the bottom end. That bottom end in our example is what's called the leading strand. The leading strand of the DNA means that that's the one that goes from 3' to 5'. The leading strand is the easy strand. It works just like we said. Topoisomerase relaxes it, Helicase splits it apart, and it scrolls through, and DNA polymerase just goes through and goes new base, new base, new base, new base, and just keeps putting a new base on there. It gets a little bit more complicated, though, when we talk about the lagging strand, because the DNA polymerase can't roll through in the same way on the lagging strand. There's always one, and leading means that's the one that's first, and lagging is the one that's lagging a little bit behind. And it doesn't move it in one smooth chunk. Instead, it's doing it in little parts. You'll see these little fragments here. Okay, these are called Okazaki fragments. And so it's done, the matching the base pair still happens. DNA polymerase still does that. But it happens in chunks instead of one continuous strand. And then those strands have to be added back in together. So I'm going to talk real briefly about the enzymes that are involved in that. So in the lagging strand, the DNA primase has to read the original strand, okay? So that's the strand that was originally there. That's the 5' to 3 strand. That's always the lagging one. Okay, so DNA primase is going to read it and say, this is what base pair is coming, okay? The DNA polymerase would then come and bring in a matching pair, okay? You'll see that it's in one single chunk right here, all right? And then there's another enzyme called DNA ligase. That's what this thing is. That DNA ligase is then going to bond the Okazaki fragments together, okay? And so they're in little chunks, and then they're going to essentially get that gap that's between them is going to get bridged by the DNA ligase. It's basically going to fill it in and link them together, okay? That's what the ligase does, okay? So basically the same. Sorry, I didn't mean to jump forward there. Basically the same as the other strand, just a little bit more complicated, a few more enzymes that are involved because it can't get done in one smooth piece like it can on the leading strand. Now, the way that this actually happens is, it's not like it takes the end of the DNA and it just unwinds it and it goes from there. This actually happens right in the middle of the DNA. And what happens is an enzyme will basically start the activation. It'll start in one section. Okay, and this is the new DNA being formed, and it happens in what we call these little bubbles. And you'll see these bubbles appear in all different places on the DNA strand. Okay, it's not just happening in one place. It's not just happening here at the end or anything like that. It's going to happen in multiple places, and so you're going to get these bubbles here. These bubbles are going to expand, okay, and get bigger and bigger until eventually you've got the whole thing taken care of, and you get what we have down here at the bottom, which are the duplicate strands of DNA. So now at the end of the process, what do we have? I've got 2 strands of DNA. Here's one, okay, and here's the other, and they are exactly identical to each other. Here they're different colors, but they've got the identical base pairs now, and then those strands could get pulled apart in mitosis and go to different cells. Okay, so that's the basic process of replication. I would say that you definitely want to run through this a couple of times, and we'll definitely do some base pairing activities in class to go over that part. But you need to run through and know each of those enzymes that's involved.